Welcome to Let's Crack NEET UG English. And okay, so first thing first is the audio check, which we normally do. So this is what we are going to check first. And we are live on the channel Let's Crack NEET UG English. And you can see the topic that we will be talking about today, that is photosynthesis. And in photosynthesis chapter, we'll be talking about chemiosmotic theory and then the factors affecting photosynthesis. This is our second last lecture on photosynthesis. That means tomorrow in the next lecture, we'll be finishing this chapter and we will be having a quiz. And before the quiz, we'll have the doubt solving session. So in case if you have any doubt, you can post your questions. So I can see few students who are already live. So just let me know whether the audio is clear or not so that we can get started. Hiren Sharma, good morning, Hiren. Is the audio clear? Rahul, good morning. How's everyone doing? Audio is clear. That's very good. Everything is clear, all clear. Dr. Vikas Sahu. Okay, Hiren, are you joining for the first time? Because I don't remember seeing your name earlier. Hi Kalai. Yes, okay. So see, those students who are like live on a daily basis, now I remember their names. So when I see a new name, then you know it comes to my mind that are you there or for the first time? So how come you joined so late? This chapter is almost complete. Just one more lecture left after today's lecture. And the other students who are already there, have they revised everything what we have done so far? Good morning, Sridhar. Oh, you were not aware of this channel. Okay. So now you know about this, and this is one channel where you know I'm going to be taking lecture almost every day. There are other channels where the lectures are taken on alternate days or maybe 3 times in a week, but this is one channel where I will be taking the lectures on a daily basis. And as soon as we are done with photosynthesis, the next chapter which I'm starting here on this channel is respiration in plants, that is cellular respiration, in which we'll be talking about all the steps like glycolysis and Krebs cycle and all those things. Good afternoon, Jayant. Which pigment is water soluble? Anthocyanin. Anthenoids, Kunal has written. Okay, so Hiren, you follow me. Very good. All right, so audio is good, and there are students who are already logged in, they're live with me, so we can get started. And as I told you, I'm going to be starting a new chapter as soon as this one is over, and that chapter is respiration in plants. So in tomorrow's lecture, I'm going to share the schedule for that chapter also. So we will be having a doubt solving session and a quiz session tomorrow. So come prepared in case if you have doubts, we'll take those up first. The thing which we will be discussing today, chemiosmosis, is one which is very, very important. And I don't know for what reason I have seen that students find difficulty in understanding this. I'll ask this question again. How many, let me ask you this for the first time. How many of you have read chemiosmotic theory? And how many of you find this chemiosmosis difficult? Photo respiration number of ATP and NADP has to be remembered. See, Jayant, what happens is if you remember the cycle, you automatically know in which step phosphorylation or ATP is required, in which particular step how many ATP is required. So if you remember the cycle, you automatically know. You don't have to mug it up. Kunal is asking, ma'am, whole plant physiology be completed by you? I am going to try and do that. Bibhash is asking about the notes. See, what kind of notes? Are you looking for the same PPT which I'm using here where I draw things and I write things? If yes, then those PDFs will be available to you once you install the app. And that is why in every class I'm telling you to install the app. All right, we will chat in few minutes, but I was asking you a question and you have not replied to that question. That is, how many of you have done this chemiosmotic theory earlier? Because the students who are in 12th right now or are droppers, they have definitely done this. In blue-green algae, photosynthesis takes place in blue-green algae, there are thylakoid-like structures in the outer part of chloroplast. Hiren says, I have. Jen says, me. Okay, Kunal, no. Hiren and Jen, those who have read this chemiosmotic theory, is it absolutely clear to you? Is there any doubt? Because the same question we will be talking about towards the end of the lecture. And this will help me prove the point that biology concepts need to be understood. Okay, so Hiren says understood. Jayant is not very sure, so no. And others have not. Ma'am, please make a book for ecology. Okay, whenever I take ecology chapter, we'll do that. Okay, so I will be asking about this chemiosmotic theory question again. Let us go over some basic things which we have to do on a daily basis. This is our schedule for this channel where we are right now. We are on 22nd and this is what we will be talking about. And tomorrow, that is 23rd, is our last lecture for photosynthesis. So there will be doubt solving and then we are going to have a quiz session. I'm planning to have like about 30-40 questions. We will see how many maximum we can answer. This is another channel and this is going to end today because this is the last lecture of mine, but if you want you can join this. This is at 12:30 and here I'll be talking about cell cycle and conducting system and all those things. So if you want to attend that, then you can do that also. This is the third channel on YouTube, Biology 101. Here we are discussing digestion and absorption, human physiology. So you can join in on this channel also. Right now we are on Let's Crack NEET UG English, which is a pure English chapter. The students who are new with me today, for them, I am your biology teacher, Dr. Neela Bakore, and I'll be taking the biology part, botany as well as zoology. Now this is the app which I was talking about. In case if you want the PDFs of these lectures, you need to install this app. So once you install this app, you get access to those PDFs which are like there after the lectures. There will be daily live classes. You can view them live and then record it. You will have unlimited access not only to my live classes but the classes of other teachers who take physics, chemistry, and there are many top educators who are taking classes there. This is one thing which I've been telling you for last couple of days, and today is 22nd, so 22nd, 23rd, these are the 2 days when this pricing of subscription Or you can say this subscription plan is available for you, because from 24th there is going to be a price increase. So if you want to avail or take advantage of this lower pricing, you can subscribe in 2 days, that is today and tomorrow, and if you use this code, that is Neela, this is my code, you will get 10% discount. This is the price which is going to be applicable from 24th. So if you take subscription after 24th, you will be required to pay little more. Alright, so this is our topic which we will be starting with. Let me see how many more students have joined. Ma'am, can we find previous lectures of genetics on app? Yes. Have you installed the app, Sridhar? And Priya has joined, so good morning, Priya. And without wasting time, let us get started with chemiosmotic hypothesis. Now, this chemiosmotic hypothesis is actually the process by which ATP is synthesized. ATP synthesis, normally whenever we talk of this process, takes place due to a proton gradient. Due to proton gradient. Not only in chloroplast but also in mitochondria, which we will be talking about in the next chapter. Here in chloroplast, when non-cyclic photophosphorylation is taking place. I'm going to just give you a brief thing of non-cyclic. It starts with PS2. A pair of electron gets excited. It is accepted by the primary electron acceptor. Then it passes through 3 molecules which are called, or which form the electron transport chain, and the middle one which we made was the cytochrome complex, and then this electron pair goes to PS1. Again that pair gets excited because of the energy absorbed. It will be accepted by the primary electron acceptor, and then through one molecule it is given to NADP. To give back those lost electrons, there is photolysis of water which takes place. So photolysis of water releases a pair of electron, a pair of proton, and half O2. So this electron is given to the PS2 to make up its loss. The proton pair is given to NADP to synthesize the reducing power, and oxygen is given out as a byproduct, right? This was our non-cyclic photophosphorylation. Now, where exactly is this non-cyclic photophosphorylation taking place? That we need to draw first. So I am drawing the thylakoid membrane where exactly this thing is taking place, and I am making only one thylakoid, little bigger, so that we are able to see all the changes. This is the thylakoid membrane, okay, and here will be another membrane of thylakoid, or rather here will be another thylakoid, and here will be the third thylakoid. So that if you're talking about the stack, appraised part and non-appraised part, okay, this is what we have to remember. So now I'm going to place the non-cyclic photophosphorylation in this membrane. So PS2, that is this one, it is located in the appraised part. Appraised means the membrane part which is in contact with the other membrane part, and PS1 is located in the non-appraised part. So here is, and we will not be drawing the whole thing in detail, okay. So this is PS2. Okay, Kunal is saying, ma'am, thoda bada likho. Okay, so let me write it a little bigger. I'm going to erase this completely. and make even bigger diagram for you. Let me make the thylakoid first. And we have to just remember that there is one more thylakoid here, so that we know which is the appressed part and which is the non-appressed part. Is it better now? Okay, Kunal was concerned about the font size, so I'm writing it bigger now. And Kalai says extremely sorry because right now can't watch, but you will watch. I guess there is some urgent work, that's why. Alright. So don't forget to watch this video because this particular concept is very, very important. And now I'm drawing the PS2 and 1. PS2 is in the appressed part. So let us make this PS2, from where this photosynthetic process or non-cyclic photophosphorylation is going to start. And PS1 is located in the non-appressed part. Okay, now let us draw the primary electron acceptors of both. Here is the primary electron acceptor of PS2 and here is the primary electron acceptor of PS1. Exactly same non-cyclic photophosphorylation pathway is what we are drawing here. Now when sunlight falls on these pigments, what happens? A pair of electron gets excited and it is accepted by this primary electron acceptor of PS2. And then it is going to pass through the electron transport chain. Again, if we want to draw it a little bigger, let us do one thing. Let me just draw it even bigger. This is the reaction center chlorophyll, PS2, and here I'm going to draw PS1. This is the primary electron acceptor, this is the primary electron acceptor. Now when the photons are absorbed, then a pair of electron gets excited. This electron pair is going to pass through the electron transport chain, and this red box which I have drawn here is the cytochrome complex. What is outside here around this thylakoid? Around the thylakoid is present stroma. And in stroma are various ions, like protons are there, various ions are present, protons, many protons are there. Now the cytochrome complex is actually a proton acceptor. It is an electron transporter and a proton acceptor. So what is meant by these two words? Transport is it takes the electron from the previous electron acceptor, that is plastoquinone. So it takes from plastoquinone and then it passes to plastocyanin. This is electron transporter. So cytochrome is in the middle. It takes the electron from plastoquinone and then passes it to plastocyanin. But as cytochrome, it is a proton acceptor. From the stroma, it has accepted a proton. That means it is holding on to a proton. You have to imagine a situation that suppose we are holding something in our hand, and when we pass on another thing from one person to another person, suppose some energy is released, we get some kind of a current or something, then what will happen? Whatever we are holding, that thing is going to fall off. Similarly, proton is with cytochrome. The cytochrome is holding on to this proton. When that electron passes through this cytochrome, energy is released. And because of this energy, the proton which was held by cytochrome is lost. The one which was held by cytochrome, now it is lost from cytochrome. From where was that proton held? The proton was taken from stroma. But when it is released inside the lumen. So this part is the thylakoid membrane, and the space inside is called the lumen of thylakoid. So what has happened so far? So far, cytochrome, which is a proton acceptor, accepted a proton. It got attached to a proton, and that proton was taken from stroma. Cytochrome is also an electron transporter. So when it passes that electron through it, it took the proton from, this was plastoquinone, this is cytochrome, and this is plastocyanin. So when cytochrome takes the electron from plastoquinone and passes it to plastocyanin, that energy which was with the electron is lost, released. Because of that energy, that proton which was attached to the cytochrome is released. Where is it released? It is released inside the thylakoid membrane, that is in the lumen part. So now what has happened is, this proton from the stroma has entered or reached the lumen of thylakoid. Now this electron will be given to PS1. From PS1, the pair of electron will again get excited. It will be taken by this primary electron acceptor. Then it goes through ferredoxin, and finally this electron will be given to NADP. Where is this NADP present? It is present in the stroma. Okay? And on the outer side of the thylakoid membrane, on the outer side of thylakoid membrane, here is present an enzyme, and this enzyme is called NADP reductase. Okay? So finally, the electron pair is given to NADP. This electron pair has reached here. From PS2, electron was lost. We are talking about this. How do we replace that lost electron? Photolysis of water has to take place. So where does photolysis take place? Water splits inside the lumen. Reason? The enzyme which is required for this photolysis is present on the inner side of the thylakoid membrane. So this is the enzyme for photolysis. It is called photolytic enzyme. And because of this enzyme, water now splits inside the lumen. It releases a pair of electrons, it releases a pair of protons, and it releases half O2. So where is oxygen formed? It is formed in the lumen and then it diffuses out. What happens to the electron? The electron is given to PS2. So that loss is made up. What happens to the proton? So the proton is inside the lumen. Now there are two sources from where the proton is coming into the lumen. One, due to photolysis of water, and two, due to electron transport chain. So whenever electron passes through cytochrome, it releases the energy. And by using that energy, a proton pair is sent in from stroma into the lumen. So proton is coming from the stroma through this cytochrome, and protons are also coming by photolysis of water. So inside the lumen, concentration of protons is increasing. Protons would tend to move from higher concentration to lower, and this is what is a proton gradient. Gradient is higher to lower or lower to higher. So here proton gradient is created. Higher proton concentration inside the lumen and less concentration in the stroma. Now these protons have to come out. They can come out only through F1, F0 particle. This particle is called F0 and this is called F1. We have talked about this particle when we talk of mitochondria. But in mitochondria, the structure is different. It is not exactly like this. So there in mitochondria, we call it F0, F1 particle, we call it oxysome, we call them elementary particle. Here we call them only F0, F1 particles. Okay? Now, up till now, is there anything which is difficult? Inside the lumen, concentration of proton increases, written by Hiren, which is perfectly right. Hiren says no ma'am, that means there is no problem. Anybody who has any difficulty so far? Because there are many questions which are asked or which can be framed only on this chemiosmotic theory. It's all clear? Very good. Priya also says it is clear. Now let us come to no difficulty ma'am. Rahul, very good. So now let us come to this proton part. How is the proton going to come back to the stroma? Why were the proton inside? One, some protons were released due to photolysis of water and some protons came in due to this electron transport chain. Which is that molecule? The molecule is cytochrome. Cytochrome is a proton acceptor and an electron transporter. Now this F0 particle, it binds with that proton which is inside and brings it to F1 particle. How is it going to bring? It moves through the membrane, it comes here and here. So every time this F0 particle is holding on to this proton, 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 and here it releases that proton. So this F0 particle is acting as a carrier molecule. It is carrying that proton. Is there any energy used? No energy. So what name should be given to this process? F0 particle carrying this proton. What name should be given to it? Is there a carrier molecule involved? Answer is yes. Is there any energy involved? Answer is no. That means it is a passive process. So movement of ions or solute passively is diffusion. But because it is with the help of a carrier molecule, it is facilitated diffusion. So now this proton which comes to F1 will pass through F1 particle, and here it is going to release that energy from higher concentration gradient to lower gradient. It is moving, and this proton comes out into stroma. But when it was going through F1 particle, that energy, see, I have drawn the arrow half part zigzag and half part straight. So zigzag line shows that there is some energy, and in F1 that energy is released. This energy is used to add inorganic phosphate to ADP. And if inorganic phosphate is added to ADP, what will be synthesized is ATP. Synthesized, that is chemiosmosis. And is ATP synthesis due to this proton gradient? Now, when we talk of non-cyclic photophosphorylation, at cytochrome, all this is taking place. And we won't be able to draw this whole thing here. And that is why what we say is, whenever electron passes through cytochrome, a proton will be coming into the lumen. When this proton comes out, ATP is synthesized. So we directly write that at cytochrome, ATP is synthesized by adding inorganic phosphate to ADP. This is what is chemiosmotic theory. Where is photolysis taking place? In the lumen. Why so? Because the enzyme which is required is here, and don't forget, for this process manganese ions are required. Where is NADP reductase? It is on the outer side of the membrane. So now these protons which are here, all these protons, this proton with the help of NADP reductase will be taken by NADP, and NADPH2, that is reducing power, will be synthesized. So where is NADPH2 produced? In the stroma. What is the reason? Because the enzyme is present on the outer side. Outer side means the side which is in contact with the stroma. Where is photolysis taking place? In the lumen, because of the enzyme. Where is oxygen released? Oxygen is produced in the lumen. What is the function of cytochrome? It is an electron transporter and a proton acceptor. So because of two processes, that is electron transport chain and photolysis of water, there is accumulation of protons in the lumen, and that results in the proton gradient formation. Protons can come back to the stroma only through F0 F1 particle. F0 particle acts as a carrier protein, brings that proton to F1, and then through F1 this proton comes out. When the proton is coming out through F1, it releases that extra energy, and that energy is used to add inorganic phosphate to ADP, and this results in formation of ATP. But in short, when we talk of non-cyclic photophosphorylation, at cytochrome we say ATP is synthesized, because we cannot draw this whole thing at cytochrome. Clear? This is chemiosmotic theory. Okay, so tell me whether this is understood by everybody or not. If there is any doubt, then I am going to take that particular part again. Kunal has written passive and facilitated diffusion, we call it, okay. So is chemiosmotic theory clear? Rahul says clear, ma'am. Hiren says crystal clear. Anything else? Anybody? Any doubt? Any point? So anyone wants me to go over it one more time? Clear, Priya also says. Divya, are you a new student? The questions which are asked on chemiosmotic theory are like the site of oxygen production, the location of NADP reductase, the source of protons in the lumen, function of F0 particle. Reshma is asking, what is this oxygen evolving complex? It is a protein which is on the inner side of the membrane and it helps in splitting of water molecule. It can be called oxygen splitting complex, it can be called photolytic complex. A quick recap would be appreciated. Okay. So what I'm going to do is, I have kept this page blank for you. I knew that somebody is going to ask for a recap. So when we do a recap, let me do it in a way which is the complete thing redrawn. But this is going to be a little quick. Okay. I am drawing the thylakoid membrane. The shape has changed, but that is okay. Now I'm going to place the non-cyclic photophosphorylation pathway in this. So this is PS2, this is PS1. This is the primary electron acceptor of PS2, this is primary electron acceptor of PS1. This part is called the membrane of thylakoid and the inner space is called the lumen of thylakoid. Outer area is stroma, and in stroma there are protons. On the outer surface there is an enzyme, and this enzyme is called NADP reductase. On the inner side of the membrane also there is an enzyme which is photolytic enzyme. Okay. Now when sunlight falls on these chlorophyll pigments, so there is sunlight which is falling on chlorophyll, here is sunlight, here is sunlight. Whenever there is sufficient energy which is accumulated in the form of photons, a pair of electron gets excited. This pair of electron passes through three molecules: plastoquinone, cytochrome, plastocyanin. So we are drawing only one, and that is cytochrome. And then the electron pair goes to PS1. When the electron passes through electron transport chain, it releases the energy. Why is the energy released? Because electron is moving from higher energy level to lower energy level. So that extra energy which it has will be released, and that is released at cytochrome. So here you can make an arrow which shows zigzag arrow. That indicates energy. Cytochrome is actually a proton acceptor. So this cytochrome has actually accepted a proton. It is attached to a proton. When this energy from electron is released, the proton which was attached to cytochrome is also released. The proton was attached from the stroma side, but that proton is released on the inner side. When is the proton released? When electron passes through cytochrome and releases the energy. So protons are now coming into the lumen. Let us complete this part first and then we will come to the photolysis. So this electron pair which has reached PS1 now again gets excited, comes to its electron, primary electron acceptor, then it goes through one more chain, that is ferredoxin, and then this electron pair is accepted by NADP. And where is this NADP? This NADP is present in the stroma. So far okay? Now, to replace the lost electrons from PS2, there is photolysis of water. Water is going to split in presence of manganese ions, and it splits into three parts: a pair of electron, a pair of proton, and half O2. This oxygen which is produced is the byproduct. It diffuses and is lost. Electron pair is given to the chlorophyll of PS2. So the loss is made up. The protons are going to remain in the lumen. So now there are two sources from where these protons are coming. One, from the cytochrome when the electron passes through cytochrome, and second, by this photolysis of water. How can these protons come out? And why should they come out? Because inside the lumen there is a concentration of proton which is increasing. So this results into production or generation of a gradient. Higher concentration of protons in the lumen as compared to the stroma. And how and from where can these protons come out? These protons can come out only through F0, F1 particles. So this particle is F1 and this particle is F0. What is F0 going to do? F0 binds with this proton and brings it to F1. So it binds with this proton, it comes here, the proton is attached, the proton is attached here, and finally that proton is released. The proton comes out through F1 particle and releases that energy which it has. And this proton is out in the stroma. The energy which is released by this proton when it was moving from higher concentration gradient to lower concentration gradient, that energy is used to add inorganic phosphate to ADP, and ATP is synthesized. Why was this proton able to come into the lumen? Because of the energy. From where was the energy released? From the electron. How come the electron got that energy? Because the photons were absorbed, and photons are nothing but the energy from sunlight. So that energy from sunlight has been converted into chemical energy, and that chemical energy is in the form of ATP. So now these protons are back in the stroma. Some protons, they are all around. So now this proton and NADP which has already received the pair of electron would receive the protons, a proton pair, and will change into NADPH2, and this is the reducing power. So as a result of chemiosmosis, ATP is synthesized, and this ATP is actually due to a proton gradient. So whenever there is an ATP molecule synthesized, there has to be a proton gradient. Now how that proton gradient is built up, it is different in photosynthesis and in cellular respiration. So what we are talking about is the proton gradient in photosynthesis, and due to which ATP is synthesized. This process is known as chemiosmosis or chemiosmotic theory. Okay, Hiren is asking, the oxygen released here is the oxygen we use to consider when writing the equation for yes, because the general combined equation which we write is carbon dioxide plus water gives you carbohydrate plus oxygen. But when we understand the complete process, we know oxygen is released during light reaction and carbohydrate is synthesized during dark reaction, but the summed up reaction is the oxygen given out as a byproduct. So now is it clear after the recap? And now you know the location where NADPH2 is produced and why, where is oxygen produced and why. So any doubts in this? ATP is synthesized here, then mitochondria is called powerhouse. See what happens is, in mitochondria the ATP which is synthesized is used for all the processes. ATP which is synthesized in chloroplast is for dark reaction. It is not for all other activities to take place, and that is why only mitochondria is called the powerhouse, not the chloroplast. By redox, if something is getting reduced, something else is definitely getting oxidized, because oxidation reduction is always together. Any other question in this? Now what you are going to do after this lecture is, you will read the chemiosmosis part from NCERT, and the same thing has been explained in bookish language. So you read that, and I'm sure this part will be absolutely clear to you. So we have done the recap also by drawing the same thing again. Those who said they were not able to understand it in the beginning, now is it clear to them? I think Kunal was one who was saying that this chemiosmosis was not clear. So is it clear to everyone now? And if yes, then we will come to the next part of this. Kunal, clear? Very good. Hiren also says okay. Now the next part which we have to talk about is the factors which affect the rate of photosynthesis. Now there are factors which I have written down here, and we are very clear about these factors. We know light is essential. Now in light, what is essential? Is quality of light important? Is quantity of light important? And is duration of light important? So factor is light. What is meant by quality? Quality means which wavelength. Which wavelength is important? We know it is blue and red wavelength where the photosynthetic process is maximum. So if you provide plant with blue-red, then rate of photosynthesis is going to be higher. If we provide, say, green light, will there be photosynthesis? Will there be photosynthesis if a plant is exposed only to green light? Okay, can anybody answer this? If green light is provided, then will photosynthesis take place? Rahul says no. Hiren says the rate will be low. Dwarkadish says no. Apurva says no. Kunal says no. Photosynthesis will take place initially, but at a very, very low pace. You remember? We talked about reaction center chlorophyll, and there were accessory pigments and carotenoids. They will absorb that green and they will collect those photons and will pass on those photons to chlorophyll A. But when all the wavelengths are available, all pigments absorb according to their capacity. Here only green is available, so only carotene or xanthophyll, they will be absorbed. So the number of photons absorbed will be less. So if green light is there, rate of photosynthesis is going to be very, very, very low. So Hiren has written that the rate is going to be very low. Is quantity, when we say quantity, it is basically intensity. High intensity, low intensity. Intensity of light is we're talking about. If very high intensity of light is available, rate of photosynthesis will start to decline, because at a very high light intensity, temperature is also going to be high, and we know if temperature increases, plants will show photorespiration. Carotenoids carry red pigments, then how green? If the pigment is red or orange, that means it is reflecting red or orange and absorbing the other wavelength. So if you remember absorption spectrum, then you will be able to relate to this. Hiren says at very high intensity, stomata will close. Yes, because as the temperature increases, it is a stress situation for the plant and the stomata will close. So moderate intensity is good. Duration, normally 10 to 12 hours of light. If available, then moderate to good rate of photosynthesis can be seen. Carbon dioxide is the next factor. Now, if concentration of carbon dioxide increases, what will happen? If you remember, when we started this chapter, I talked about Blackman's law of limiting factor. I'm going to go over it because probably some of you did not attend or see that lecture. So it is Blackman's law of limiting factor. According to this law, the rate of process, in this case we will talk about photosynthesis. Rate of photosynthesis is regulated or decided by the factor which is in minimum concentration. And there are four things which are required by plants for photosynthesis. One is water, second is carbon dioxide, third is light, and then chlorophyll or the pigments. Light is available in plenty, chlorophyll is anyways present inside the leaf, and assuming that you have watered the plant, carbon dioxide concentration in the atmosphere is very low. So this factor is going to be very low. So if concentration of carbon dioxide increases, the rate of photosynthesis will increase, but for a short period of time, only for little increase in concentration. So if a rate of photosynthesis increases, that means there will be more organic matter synthesized. So what happens when you add fertilizers to the plant? When we add fertilizers to the plant, the production increases. And production means carbohydrate is more, starch is more, seed size is bigger, fruit size is bigger. So same thing is happening here also. So this is called carbon dioxide fertilization effect. Very important. But if carbon dioxide concentration increases beyond a limit, this is one part. Now if concentration of carbon dioxide increases beyond certain limit, then it has toxic effect. Then it has toxic effect. What happens in toxic effect? That extra carbon dioxide is going to react with water to form carbonic acid. Carbonic acid is going to dissociate into H plus and bicarbonate ions, and when H plus increases, it results in change in the pH. The pH becomes acidic, and acidic pH lowers the rate of photosynthesis because it results in closure of stomata. Stomata close. So little increase in carbon dioxide is beneficial because photosynthesis will increase, and beyond certain limit, if carbon dioxide concentration increases, then it is going to have a toxic effect. The third factor is oxygen. If concentration of oxygen increases, it results into photorespiration, and this is known as Warburg's effect. So oxygen is, if it is little more, then how, Rahul is asking a question, how aquatic plants like hydrophytes get carbon dioxide? It is dissolved in water. The way they take oxygen, the similar way they take carbon dioxide. Hiren has a question. Concentration of carbon dioxide will also result in greenhouse effect. Yes, temperature increases and that will result into closure of stomata. Very good. Carbon dioxide has high solubility in water. It is enough for the plants to be absorbed, and then photosynthesis can be done. Water is essential for photosynthesis because it is a raw material. If water is not there, then that replacement of electron will not take place. Temperature is again very important factor. At high temperature, there are two things. One, rubisco will act as oxygenase and will cause photorespiration. And at high temperature, it is a stress situation. To minimize the rate of transpiration, stomata close. And if stomata close, then there is no exchange of gases. Then there are some internal factors also. Internal factor means amount of chlorophyll. how much chlorophyll is available. So 1 gram of chlorophyll can perform how much photosynthesis, and that is called assimilation number. Okay, so these are some internal factors. Internal factors would be arrangement of chloroplasts, number of chloroplasts. This is like inside the leaf. But most of the factors which affect the rate of photosynthesis is actually external. So all these factors that we have talked of, they are external factors. And all these factors like light, carbon dioxide, oxygen, they play a very significant role in deciding the rate of photosynthesis. Some factors increase the rate of photosynthesis, some factors decrease the rate of photosynthesis. Water is raw material. Without water, those electrons will not be replaced, and if electrons are coming out of the chlorophyll of PS2 or PS1, then ultimately the chlorophyll molecule structure is going to change. So that will again affect the rate of photosynthesis. And temperature anyways is again very important because it changes the behavior of enzyme rubisco, and high temperature is a stress situation for the plant, and during this the stomata will close. So this is what I had planned for today, and I hope you have understood both the things, that is chemiosmotic theory as well as the factors. Now when we meet tomorrow, same time 11 o'clock, we will be talking about first the doubts that you may have, and then I have planned a quiz for you. So read everything carefully from NCERT, from the notes that you have taken while I was teaching, and don't forget to go for this subscription. Take the subscription as early as possible, because I have shared that sheet with you which shows that there is going to be increase in price, and if you want to avail this 10% discount, don't forget to use my code, that is Neela. Alright, so is everything clear what we talked about in today's class? Before we wind up today's class, just let me know. Hiren, thank you. Have a lovely day. You too. Have a nice day, and don't forget to read NCERT, especially the chemiosmosis part. Okay, I am not able to read this name clearly. Wafola. Ma'am, then how does a plant living in high temperature manage to have photosynthesis? They are C4 plants, or they go with CAM, that is crassulacean acid metabolism. We have talked about this in the previous class. Okay, so Hiren, Sridhar, Rahul, everything is good. Alright, so go over all these things, revise the complete chapter so that you are able to answer the questions on the quiz tomorrow. Okay, so I will see you tomorrow, same time. So till that time, take care and have a nice day. Bye-bye.